today we're going to have another, in fact, the last one of our disease-specific lectures on HIV/AIDS, which of course is a very important and critical health problem in today's world. So while this is a very large topic, what I'm going to touch on are some important areas, and then also give you some places to go in the event you want more information. So the question is, what is HIV/AIDS? Well, HIV stands for the human immunodeficiency virus. And the virus is, of course, what is responsible for AIDS, which is acquired immune deficiency syndrome. And again, this is a epidemic disease, and it's currently being controlled quite well by these HAART, H-A-A-R-T, medications that we're going to talk about later in the lecture. But we'll see that even though the incidence is certainly continuing, right? People are continuing to develop AIDS. Once they get AIDS, the hope and the length of life now that's available for people who develop AIDS has greatly improved in the last 10 to 20 years. So the thing is, once the individual is infected by the HIV virus, basically the virus is going to be there the rest of the person's life. There are a couple of important exceptions to that. There's one individual in Berlin that was successfully able to completely eradicate the virus, and we'll touch on that later in the lecture. And there was another baby recently born in Mississippi, but that baby actually has now a very low level of virus, so we'll again talk about that as well. So HIV is transmitted through body fluids, and it's a type of virus. So the HIV virus actually lives in a T cell, the CD4 cell, and it infects and lives, of course, in the immune system. And because of that, there is a characteristic immune suppression that occurs with individuals who have AIDS. And often it's not necessarily the AIDS disease that causes mortality. It's actually the other opportunistic diseases that occur because of the immune suppression. And we touched on a couple of these diseases in our other lectures, and I'll bring them back to recall, to provide some connection to those lectures, as well as talk about a new, or not a new, but at least another opportunistic infection, the Kaposi Sarcoma. So HIV can be transmitted through sexual contact, through injection drug use, pregnancy, childbirth, occupational exposure, and what's meant by that are exposure to body fluids, and blood transfusion and organ transplant. Now, in the beginnings of the HIV epidemic, this blood transfusion actually was a significant method for disease transmission, because at the time the virus was not identified, the antibodies couldn't be measured, and individuals who got blood transfusions from donors that had HIV or were infected by HIV would then develop a very extreme form of AIDS. But now that we have the AIDS test available, now that we can evaluate for the presence of antibodies, then of course our blood supply has been screened for all the potential people, high-risk individuals who might donate, but also the blood is screened after donation to make sure that the AIDS virus is not there. So what is the very close to being current adult prevalence, adult HIV prevalence across the world? Well, the highest prevalence country is Africa, and other high prevalence areas, of course, are the United States, North and South America. India, which I'm going to talk about later on, is a location where I in particular am conducting research with my colleagues. And even though that's potentially, and in fact, a lower prevalence country than Europe or the United States, for instance, the number of people who live there make India actually the contributor of the third largest number of cases in the world. So the reason why I'm pulling up this figure is also to show the concentration of AIDS cases in the United States. But in the epidemiology lecture, we also bring this point to light as well. And you can see that Los Angeles, San Francisco, Miami, and New York City are high prevalence areas, and these areas are also responsible for a large number of cases in the United States. Now you might ask, well, what's the reason for that? Well, the reason for that is there are also a large number of gay men or men who have sex with men in these areas, and they are the biggest the highest risk category in the United States. So I just want you to look at here, looking at, for instance, Los Angeles compared to San Francisco, and you can see that even though San Francisco is a denser area, there are more people per square foot living in San Francisco than Los Angeles, for instance, and also the prevalence of HIV is higher in San Francisco than Los Angeles. Los Angeles is responsible for a larger number of cases. And this point is actually important in relationship to a country like India, which we saw had a lower prevalence than the United States, for instance. However, because more people live there, India actually produces more cases. So this is a very important point, because I think if you only look at prevalence, then you miss the phenomena that potentially these lower prevalence areas can certainly produce a larger number of HIV and AIDS cases. So here are some pictures, some frequencies of different methods of disease transmission, and you'll see that they differ greatly in males and females. Now, in males, male-to-male sexual contact is responsible for 67% of the transmitted disease of HIV and AIDS. In women, of course, high-risk heterosexual contact is the highest risk category. Injection drug use is also another prominent category, as is in men as well. And in men, we also have the joint category of male-to-male sexual contact as well as injection drug use. Now, remember when we talked about the malaria, we talked about vectors, we also talked about parasites, that one of the ways, right, the major way to get infected by a blood-borne pathogen is through exchange of blood. So in relationship to malaria, for instance, we have the transmission of malaria occurring by an insect vector. Now with HIV and AIDS, what we have there is we have sexual transmission, and we also have transmission by exchange of bodily fluids. So both of those methods are really the primary way in which HIV virus is transmitted between individuals. Now, in the United States, this is the primary way in which disease transmission occurs. In other countries, it's different. In Africa, the main way of disease, or the primary method for disease transmission, is through heterosexual contact. So, how does the HIV virus integrate itself into the T cells? Well, HIV is called a retrovirus, and the reason for that is it's actually an RNA-based virus. And remember, RNA is copied from DNA, but with a retrovirus, what we can do is go in the opposite direction. So we start with RNA, and then we essentially copy back DNA. So it's a complementary copy going in the reverse direction. And then we get viral DNA, and that viral DNA integrates into the host genome. And then when the host genome, when that particular chromosome gets replicated, then, or when that cell goes through cell replication, then we are also replicating the virus. So, you know, we can replicate the virus actually by two ways. One, through cell division of the cell itself, and the other way is through transcription and translation. Now, what do we know about the T cell? The T cell undergoes a lot of replication. So this is a very significant fact about transmission of HIV/AIDS. Once an individual gets infected, that infection can certainly spread quite rapidly. So here's another picture of that. So you'll see that the the HIV virus fuses with the cell membrane. The genome of the virus is injected, so to speak, into the cytoplasm. We get reverse transcription of DNA from RNA. That DNA gets integrated into the host DNA. And then the host DNA, as we talked about, then goes through transcription and translation and also potentially cell division. We produce then viral messenger RNA that then assembles, integrates, and translates itself, right? We get viral proteins, and then these viral proteins are then assembled into the HIV/AIDS or the HIV virus, which then leaves the cell and goes on to infect other cells. Now, the reason for this, I included this particular figure, is because it shows the places in which these HAART medications actually work. So they work in terms of protease inhibitors, in terms of fusion inhibitors, and in terms of this reverse transcription and translation. So that's very important in relationship to the drugs that are currently on the market. And it's important to know that these drugs are actually disseminated across the world. So we'll talk about that when we talk about the example in India. So here's a picture of our viral load and the changes in these immune cells. So the individual is infected here with the HIV virus. We see that right away the plasma virus load increases, and then it rapidly declines. Now, there is certainly some immune mobilization against the virus in the beginning, but you'll see here that the virus basically goes to this dormant level, and it's only here a number of years later that the virus can reemerge and then go on to develop full-blown AIDS and death. Now, the other important part of this figure is to look at the CD4 T cells, because that's the place in which, or that's the location that the HIV virus infects. So you'll see that right away the CD4 cells go down, they come back up, and they stay at this level, and then they gradually start to decline. Now, remember, in the earlier discussion, we talked about how that this virus actually infects other T cells, and those T cells eventually die off, and we get a decline in these CD4 cells. And when they get low enough, that's the time that the HIV virus let's put it that way. Let's put it this way. That's the point at which the immune system is suppressed far enough that full-blown AIDS develop and potentially mortality occurs. This point is very important also in relationship to opportunistic diseases, because as we'll see in the next slide, opportunistic diseases come on board at certain points during that immune suppression. So when the CD4 cells get low enough, that's when we see very characteristic emergences of these diseases. So that's really important. Also, what's important is that the virus evolves over time. It changes in phenotype, it changes in immunogenicity, such that what may work in the beginning in terms of treatment may not necessarily work at the end. So that's critical as well. So here is a picture of what is going on in terms of evolution of HIV, of the HIV virus in relationship to the AIDS disease. So here, evolution.berkeley.edu has come through again in relationship to this discussion. And really, they have a whole series of lectures about this whole phenomena. And it's equivalent to if we had if we had Drosophila, right, migrating to different islands, and these populations living there over time, and then depending on the conditions in each of the islands, they potentially evolve various traits to enable those particular flies to live. And eventually, they become so distinct from one another, they're not even the same species, potentially. Now, with HIV/AIDS, the same situation can occur in individuals, so that over time, the AIDS strain 1 and individual 1 may be different from HIV 2 or 3 or 4. Now, what can happen is you can have certain strains in certain areas that are more or less indigenous to that area, but within each individual, we have different selection pressures, different health, levels of health, different numbers of cells, different capacities of the immune system to, certainly fight off the HIV virus. And so we develop different phenotypes of these viral strains over time. And that, in a sense, is an evolutionary pressure that's really unique to the HIV virus, because it's a virus that's capable of change, just like influenza, for instance. That's another virus that's capable of a lot of change. So here's that graph I was telling you about, and these are all these different opportunistic illnesses down here, and they seem to have the characteristic time that they emerge based on that drop-off in CD4 cells. So, for instance, herpes simplex virus begins to emerge, and now these stars represent the mean level, so they begin to emerge at the mean of 200 CD4 cells. Herpes simplex or herpes zoster, which is shingles, tends to emerge a little lower, and so forth. We have, very characteristic diseases. We have Kaposi Sarcoma, we have Pneumocystis Pneumonia, the candidiasis. These diseases we've actually talked about in our lectures, toxoplasmosis, and so forth. So these diseases emerge at certain points depending on the degree of suppression going on with the immune system. So here's another graph that shows how the incidence of these opportunistic infections have changed over time in relationship to HIV. So in the early 1900s, we weren't using at the time these ART medications, and because of that, these opportunistic diseases had a much higher incidence, but over time, they have gone down. So pneumocystis, candidiasis, Kaposi Sarcoma, the diseases that we just talked about in the earlier graph, they have all started coming down. And it's because by use of these medications, what we're doing is we're strengthening we're strengthening the immune system, and we're strengthening the capacity of the immune system to fight off these opportunistic infections, because some of them, like for instance Kaposi Sarcoma, was very, very rare until HIV/AIDS emerged as an epidemic disease. And then actually Kaposi Sarcoma in the beginning of the AIDS epidemic was actually one of the criteria by which an individual who was infected with the HIV virus was identified. So here I'm going to go back to our fungal lecture and talk about one of the fungal diseases that emerged after the AIDS epidemic. And remember that these fungi are opportunistic. They generally do not infect healthy individuals. They're harmless, in fact, but they tend to emerge in individuals who have a debilitated immune system. Now, they don't just happen with individuals with HIV/AIDS. They can happen among individuals who are taking drugs to suppress their immune system. Remember, one of the reasons for that is if, for instance, you get an organ transplant. If you get an organ transplant, you want to suppress the immune system to make sure that the immune system does not attack the transplanted tissue. So an example, of course, is pneumocystis. That's also during the beginning of the AIDS epidemic. That was another disease that was actually a marker for the individual having or being infected by HIV/AIDS. It's one of the most frequent and severe opportunistic infections. The pathogen can be found in healthy lungs, but it usually doesn't create much of a problem. Now, if the individual is immunodeficient, however, the susceptibility is greatly increased. So here we have actually a cell histograph of the actual pneumocystis infecting the lung tissue. So remember, in our TB lecture, we talked about the co-occurrence of TB and HIV/AIDS. Now, again, in the early 1990s, we saw that a pretty large percentage of individuals diagnosed with TB in the United States were individuals that were actually also infected with HIV/AIDS. And that has gone down over time. Why? Because of the emergence of these aggressive HIV/AIDS drug treatments. Now, I also wanted to spend some time talking about Kaposi Sarcoma, because Kaposi Sarcoma was a rare cancer. It only occurred in individuals, fishermen actually who fished in the Mediterranean were actually a high-risk group for Kaposi, but basically it was just not a very prevalent cancer. It was basically a very rare cancer until the emergence of the AIDS epidemic. And so Kaposi Sarcoma is a skin neoplasm. It's vascular in nature, but it can develop a more serious stage, and it can become systemic and certainly infect any organ, actually. There are several subtypes, but they all have this human herpes virus 8. So Kaposi Sarcoma is actually a virally induced cancer. So individuals who have HIV/AIDS and Kaposi's also have this HHV8 virus as well. So in how it looks, so Kaposi Sarcoma looks like these reddened papules that are filled with blood, and these papules rapidly spread. And the interesting thing about Kaposi Sarcoma, at least the skin form, is actually treated with HIV/AIDS medications. So you can see that this is a picture of how the cancer develops, so to speak, and in which cells. So they actually develop these particular cellular phenotypes called Kaposi Sarcoma spindle cells. Now, we didn't really talk much about cancer. We did talk about tumor immunology, and in there we talk a little bit about cancer development. So one of the things that happens with cancer is you start out with normal cells, and then over time, as they become more de-differentiated, then they turn into a more aggressive, lethal form. And that's certainly what happens with Kaposi sarcoma, that they develop into this form that then this syndrome, the Kaposi sarcoma disease, can actually spread to other tissues. So here is a picture of just kind of an algorithm that basically indicates how to treat Kaposi sarcoma. So in the beginning stages of Kaposi, when it's confined to the skin and the lymph nodes, then actually the recommended treatment for that condition is with these AIDS medications. Now, if the disease is more disseminated and if it's progressing rapidly, then actually you treat Kaposi's with a chemotherapy regimen plus these AIDS medications, and then that's in the way in which you can get the disease to diminish and decline. So I brought up this figure here of India to point out that currently I'm working with colleagues at UCLA, and we were funded by the NIH to conduct a nutritional intervention among women living with AIDS in India. And so it's interesting that the study area that we are working in is Andhra Pradesh, and the part of Andhra Pradesh that's very dark right here is actually where our study site is. And it's interesting because, as I talked about earlier, the numbers of AIDS cases that come from India is the third largest number of AIDS cases in the world. And these particular high-risk areas, quite a lot of individuals develop AIDS or HIV AIDS. And as I explained earlier as well, that the WHO and the United States and other countries have basically provided India and other countries with free AIDS medications. And so the individuals in our study are all on these ART medications. But interestingly, after receiving the medication, the next most important criteria or the next predictor of risk in relationship to mortality is the quality of nutrition. And so what we're doing in India is we're providing at least some of the women in our study, it's a randomized trial with a protein formulation, because one of the things that happens when people experience immune suppression is actually protein depletion. So one of the markers for HIV AIDS or severity of disease is what's called muscle wasting. And the muscle wasting really occurs because there is such a demand for protein, because the immune system, if you can imagine, is going through a lot of cell replication and a lot of production of cytokines and interferons, all of which are protein in nature. And so there's a real protein drain in relationship to experiencing the disease of HIV AIDS. And because of that, we found that, at least in our pilot study, that implementation of protein supplementation, along with certainly care and support, that the individuals who received the intervention actually improved in muscle mass and weight, and most importantly, they improved in number of CD4 cells. And the CD4 count is one of the outcomes that are evaluated in relationship to how severe the disease is in relationship to individuals who are infected with the HIV virus. So, CD4 count is actually an important parameter that individuals who have HIV AIDS are measured over time quite often. So in any event, this is the second year of our study, and it's a 5-year study. So I just actually came back from India, gosh, about 2 weeks ago. And boy, I'm telling you, in these villages, it was amazing because we would visit the homes of individuals who actually had the disease. And, some of them lived across the street from each other or down the street. I mean, there weren't really streets in the villages, but boy, they didn't live that far away from one another. So it was very striking that the prevalence of disease, at least in the areas that we were working in, was pretty high. So anyway, good, and I just want to, leave with, just good places to go if you need more information about HIV/AIDS, because basically I'm just barely scratching the surface with this lecture. There's a site called AIDS.gov in the United States. It provides a lot of good information. The WHO provides a lot of good information as well. The CDC in the United States, various organizations in India. The NACO organization is the Indian-based government organization that studies AIDS or HIV/AIDS. And then, of course, going to the literature, as I indicated, evolution.berkeley.edu provides some very good information about the evolution of the AIDS virus. And so there's quite a lot of information out there. Much of it is freely available. And if you want more information about HIV/AIDS, I highly recommend that you go to these sites. So our first example question: why do you think Kaposi sarcoma can be treated with aggressive HIV therapies? We touched on it a little bit earlier in our discussion. Number one, improvement in immunity. Big time. That's the most important reason, because these drugs, if you saw that those various parts of the integration of the HIV genome into the host genome, that's one area. The entry of the virus into the T cell, that's another place. The exit of the virus outside of the T cell, all those three areas are targets for the drug. So if you lower if you can lower, the viral load, what happens is you then increase the CD4 count. Sorry about that. Let's make that a little. CD4 count goes up. So it's a direct correlation. If you lower the viral load, you increase the T cells. And remember, there were those characteristic places that those opportunistic diseases developed. And so if you can increase the capacity of the immune system to recognize and eradicate these opportunistic diseases, you will then A, lower the risk, but B, also be able to treat the opportunistic infection by actually improving the immune response. Because let me tell you, the immune system does a great job. In fact, the immune system does a much better job than a lot of drugs on the market. So the second example, what do you think the complications are for developing a vaccine for HIV? Number one, rapid evolution, right? We saw that in the Berkeley slide, that because the HIV virus is rapidly evolving, what part of the virus is consistent enough that you can actually develop a vaccine against it? Now, we have seen 3 diseases, right? All of which, for various reasons, are very challenging to develop vaccines against. Tuberculosis, malaria, and now HIV. But HIV is a very clever pathogen, right? It's capable of rapid evolution. And originally, the virus actually was a zoonoses. Remember, we learned about zoonoses. That was a disease that occurred actually in primates, in monkeys, and chimpanzees, it's now thought of as the species that originally provided that first viral dose, right? And the reason for that is in West Africa, it's called bushmeat, that actually the folks or the people that live in West Africa actually eat monkey meat, so to speak. It's called bushmeat, and that eating of the monkey meat is what enabled the virus to jump from chimpanzees to humans. And often what happens with zoonoses is that they're very severe. They tend to be very severe diseases. They have high virulence and pathogenicity because the human host has not developed any immunity against those diseases. So rapid evolution is the first reason for what the complications are in terms of developing a vaccine for HIV, that we have to find a consistent ligand that does not evolve. Now, one of the things that's at least happening in the United States is for very high-risk individuals, they're basically given prophylactic AIDS medications. And then what happens is, in the event they get infected, they're basically priming themselves for being able to eradicate the virus, potentially before it develops any capacity to integrate into these T cells. And it's actually becoming a more commonplace practice in the United States. In the United States, the AIDS medications are still relatively expensive, but this is potentially turning out to be not necessarily a vaccine treatment, but more like a prophylactic treatment, that some men having sex with men are actually taking these HIV treatment medications without being infected by the AIDS virus or the HIV AIDS virus, assuming that they may potentially get infected. And the idea is to be able to eradicate the virus before it gets to a point where it can actually register even. And some individuals have been found to have a very low viral level, but it's nowhere near the viral level that it would be if they weren't on these medications. So one potential approach may not even be vaccination, but maybe these prophylactic treatments. And with these treatments, potentially we can lower the virus or create a environment that is very unwelcoming for that virus, if indeed it infects the individual. So that concludes our third disease-specific lecture for the day. Thank you so much for visiting educator.com.